Hello! This video demonstration will show the proper procedure for growing a hard-water biofilm matrix in silicone tubing, as performed by the Standardized Biofilm Methods Laboratory. This procedure takes 4 days to complete, the first of which is dedicated to the collection of items and sterilization of the system. After donning the proper safety equipment and sanitizing the scale, 10 milligrams of TSB, or tryptic soy broth, was weighed and put into a 2-liter flask. To this flask, 1 liter of deionized water was also added. The flask was labeled and covered in tinfoil and set aside to be autoclaved. A length of size 16 tubing, with neoprene tubing in the middle to go through a pump, was also prepared. The final piece of equipment required for this experiment is 2 small lengths of tubing connected via a T-connector. All these items were autoclaved and left to cool overnight. Day 2 is devoted to the inoculation of the system. 2 loopfuls of the frozen stock of Pseudomonas aeruginosa 15442 was swirled into the 2-liter flask. The now sterilized piece of tubing was aseptically removed from the bin, put into the 2-liter flask, and the neoprene part was threaded through the pump. The tubing was filled with solution and allowed to incubate for 1 hour at room temperature. After this hour had passed, the pump was set to 2 milliliters per minute and set to run for 24 hours. Day 3 is devoted to introducing the hard water to the system. Each small length of tubing connected to the T-connector was placed in either hard water solution 1 of sodium bicarbonate or hard water solution 2 of magnesium chloride and calcium chloride. The influent end of the tubing was connected to the sterile T-connector, and the pump was turned on for 15 to 20 minutes so that some scaling could occur in the biofilm-coated tubing. Look at those little clumps go. After the 15 to 20 minutes had passed, the pump was stopped, the T-connector was disconnected, and the influent end of the tubing was placed back into the 2-liter flask. And the pump was once again turned on for 2 milliliters per minute for another 24 hours. The 4th and final day of this experiment is devoted to harvesting the biofilm and plating the resulting dilutions. Using flame-sterilized scissors, approximately 2 centimeters of tubing was cut from the rest into 10 milliliters of dilution water. Then, using a sterile wooden stick and flame-sterilized forceps, the biofilm inside the tubing was scraped away and swirled into the 10 milliliters. After all the biofilm had been collected, the 2 centimeters was dropped back into the water, and the sample was submitted to alternating bouts of vortexing and sonicking for a total of 5 times. This freshly mixed solution was then diluted and plated according to the drop plate method. Thank you for watching, and we hope you enjoyed this short video demonstration.